Hello everyone! Due to COVID-19 pandemic, we are not allowed to have face-to-face interaction with our students. And with that, we cannot do laboratory activities, which is very crucial to the teaching-learning process. So, let me bring to your doorstep, or not to your doorstep, but to your screen, the laboratory of the Western Philippines University. So please come to our Microbiology laboratory. This is my laboratory. Basic to microbiology laboratory is the microscope. This is where you view specimens. Of course, in microbiology, we are dealing with microorganisms and we can only see them through the aid of the microscope. This is a very simple compound-like microscope with a built-in camera that can be attached to the computer, so it's easy to visualize, capture and visualize image. Next, we have in here micropipettes. We have several sizes of this. When I say sizes, that pertains to the volume. Okay, so micropipettes are used for a very precise measurements of small volumes of liquid or chemicals, and especially when you are dealing with DNA materials. And speaking of DNA materials, we have in here the PCR, or the polymerase chain reaction. This one is able to make millions to billions of copies of DNA material so that researchers or scientists are able to visualize it. So when I say visualize, how do we do that? So first, we have to load the amplified DNA into gel, so gel wells. And that gel must be loaded in here in this gel electrophoresis. Okay, so inside, the DNA fragments, including proteins as well, can be separated. DNAs are negatively charged, and so the DNA are pulled towards the positive electrode. Okay, so from here, how do we know that we really have the amplified DNA? We need to have a picture of that. And so, we have in here the UV trans illuminator, and this is what it looks like inside. Okay, so you open this, you load your gel in here, you cover this and put this back. This one is to contain the UV light inside. Okay, you place your camera in here, and so you turn this on and capture the image. So how do you know that you have the DNA? You will see an illuminated band in the DNA, in the gel that you will see under the trans illuminator. Okay, so next is we have the vortex mixer. We are using this to properly mix liquids inside small tubes. In some instances, we are also using this for DNA extraction, as part of the process. Now, we also have in here refrigerator, another basic in most laboratories. This is where we store our samples for future use. But, of course, this time, it's empty because we do not have students. One of the topics that I usually emphasize with my students is the application of aseptic method. When I say aseptic method, it means avoiding every possible contamination. And one of the ways to achieve it is the use of laminar flow cabinet. So this laminar flow cabinet will minimize, remove as much as possible contamination. So this is where you process your samples inside so that your sample is free from possible contamination, and also you, the researchers, can be protected. Another important equipment that must be present in a microbiology laboratory is the incubator. So we have in here an incubator that's quite spacious, that can contain several samples. An incubator is a controlled environment. This is where you can control temperature, humidity, and other environmental factors that would affect growth of microorganisms. So you have to set this to what is optimum to the microorganism that you are trying to grow. And next, we have in here an autoclave. Now, my students before in Mindanao State University, do you still remember this? Oh, this is quite an old type of autoclave. We are using this connected to gas. This is still very, very useful because blackouts are still very real here in our area. So when there is no electricity, when there is no power source, we are using this to sterilize so our experiments can still proceed. Okay. Here now is a slide drying bench. We are using this when we would like to hasten the process of air drying and preservation also of slides. When we are making specimen on glass slide, this is where we dry them. This is quite old, but it still works. Okay. So, this one is our colony counter, another basic one in microbiology laboratory. So, in colony counter, if you turn this on, you have here the area where you put the Petri dish, where you have the colonies to be counted. And you have in here the pen that could mark the colonies where you already have counted, and you have in here the counter. Here, the counter. So when you count, like for example, 1, 2, 3, you press 1, 2, 3, and so on. So you don't lose counts. Okay. We have in here the top loading balance. This is where we weigh the components of culture medium, for example, and of course other samples that must be weighed precisely. Another one is the blender. So this is just a multi-purpose blender. You also have this at the home, but here in the laboratory, we are using this to homogenize samples. And here we have the microwave. Our microwave oven is not really for heating food, but we are using this to cook our culture medium. For example, the nutrient agar. So it's quite easy. It's faster as well and tidier to cook the culture medium in here rather than using hot plate or the stove. Okay. Next, we have the vertical top loading autoclave. So this is what it looks like inside. It can sterilize quite a number of Petri dishes, test tubes, and other glassware. The principle of sterilization in an autoclave is actually the use of the combination of pressure and temperature to kill microorganisms in a shorter time. For example, 15 to 20 minutes at 121 degrees centigrade. So you can kill bacteria, including those which are spore forming. Okay. So here we have our cabinet. So this one contains our culture media. We have a bit of Petri dishes in here and test tubes, and I still have lots of them hidden somewhere. Okay, so and other things also. Another cabinet we have in here. Oh, what is this for? First, we have here our first aid kit. And inside are actually, this one is actually the storage area of the materials being used by the students. And these things in here are the materials that my students were using just before the pandemic. Okay, let's close this one and proceed to our shaking water bath. Okay, the shaking water bath is also a type of incubator. So in here there is simultaneous incubation and shaking. So that is to continuously mix the sample, and also somehow it has the capacity to do extraction of biochemical components from the microorganisms that you want to extract and be tested. Okay, so this is the shaking incubator. If you look at it, see, you will see places here where you can put the test tubes. Goes. Another one here is a horizontal autoclave. Oh, this is another sterilizer. This is what it looks inside. This is horizontal, very ideal for sterilization of Petri dishes and empty test tubes. Why empty? Because you need to put them lying down so the content will not be spilled. Okay. And the one down here is the refrigerated centrifuge. The refrigerated centrifuge is actually a pair of PCR and all other things pertaining to DNA analysis or DNA experiments. In here, you can have temperatures from minus 20 to minus 40. That is very ideal for DNA, RNA, and protein analysis, or the centrifugation in that temperature that would maintain the quality of your samples. Okay, so now here is our working table. This is where my students would process their experiments, and I would always tell them to clean the surface and always, what's the word? Disinfect. That is to avoid contamination. So that's all. I hope you enjoy our mini tour in our mini laboratory. So I hope you also got something from it. Thank you very much for watching. Bye-bye!